Well, welcome to the Project ALS prosetin live stream hosted by me, Valeries Perspective, and our Erin Fleming. Wave, Erin, so people know. Today's honored guest is Dr. Emily Lowry, one of the scientists, perhaps chief among them, who took prosetin from a bright idea 6 years ago to now and here on the verge of human trial with prosetin. And now for those of you who may not know, prosetin is a new ALS drug candidate. Last year, Project ALS decided that prosetin was so promising, we were going to deliver it to human trial in 2020. Project ALS built a team of all-stars, scientists, doctors, patients, FDA experts, companies that do manufacturing and safety testing, all as one team. The team to get prosetin to human trial in 2020. Now, the purpose of these live streams is transparency of process. Plus, we're proud of the prosetin 2020 team, and we really want you to meet them and get to know them a little bit. This works both ways, ideally. We want you to ask our guests questions, ask us questions. Please just write in now and we'll take the questions. We really want to work toward making this a conversation. Now quickly, just as a recap of the three main reasons Project ALS and scientists are so excited about prosetin as a potential treatment for ALS. Number one, prosetin is brain penetrant. It gets into the brain where few drugs ever seem to make it, which is probably a good thing since ALS is a brain disease. Number two, prosetin hits a therapeutic target in the brain called ER stress. So that is to say, when prosetin enters the brain, it hits a therapeutic target relevant to ALS. Number three, we're excited about prosetin because it's a top performer. What does that really mean? What it means is that out of thousands of FDA-approved drugs for other illnesses and thousands of chemical compounds that have been tested, prosetin performed far and away better in every ALS test we have. Now, COVID has knocked the prosetin campaign back on its heels a little bit. We've lost roughly 3 months from our original schedule in delivering prosetin to human trial. But the prosetin 2020 team continues to meet responsibly, multiple times a day, per week, to minimize the loss of time on the program, but to maximize productivity. So, which is to say, quite frankly, prosetin2020.org needs you. Immediately, $5, $20, fat cats, you know, you could fork over maybe $100, but to help us keep prosetin on track, and that's prosetin, PROSETIN 2020.org. So just to let you know, Project ALS was really on goal to raise the additional $1.5 million we had estimated it would take to deliver prosetin to human trial. And we were well on our way, but then COVID struck and our revenue streams sort of slowed, and our fundraisers for these months were all canceled. Our pitch meetings with funders, potential funders were canceled. But now with you, we must take prosetin 2020 on our backs and raise the remaining $900K together. And we'll get back to how you can help more specifically in a bit, but I'm so excited for our guest. So last week on this program, we met Kevin Phelan, prosetin 2020 team member who is an FDA expert. Now let's switch gears a little. Today's prosetin 2020 team member and our honored guest today is all science all the time. He was there at the dawn of prosetin. Erin, let's meet her.
Thanks, Valerie. So I think for those of you who have been following prosetin development over this last few months, as we've been trying to keep everybody updated, you've probably already heard from our guest today, Dr. Emily Lowry. She's one of the lead scientists behind prosetin's development. But today we wanted to have sort of a more informal discussion with Dr. Lowry and really learn more about what brought her to ALS research and drug discovery to prosetin, and what the process of developing prosetin really looked like. So, you know, how did we get a drug that was brain penetrant and a really high performer in a range of ALS tests and, you know, was the top performer candidate that we wanted to bring forward. Before we get into the questions though, I just wanted to introduce Emily. Emily Lowry, PhD, is the Director of Internal Operations at the Project ALS Therapeutics Core at Columbia, or the Core, where she designs and coordinates ALS drug screening efforts and oversees the day-to-day work of the Core's entire scientific team. In this role, Emily Lowry, she develops novel approaches to ALS drug discovery, optimization, and evaluation, and she also works closely with both the Columbia scientific team and our Project ALS team to establish scientific collaborations with outside academic and industry partners. Dr. Lowry is an expert in developing and improving cellular models for ALS drug screening. And in her almost 8 years at Columbia, I think, she has validated new protocols to make stem cells into motor neurons, which are the cells that are vulnerable to degeneration in ALS. She's optimized several small molecule-based screening platforms to identify new potential therapies for ALS. And of course, that includes our lead candidate, prosetin. Dr. Lowry received a BA in cellular and molecular neuroscience from Barnard, a PhD in neurobiology and genetics from the Rockefeller University, and she conducted postdoctoral research at Columbia, first actually in the Project ALS Jennifer Estes Laboratory for Stem Cell Research, and later in the lab of Henrik Victorly, who is the co-scientific director of the Core. So without further ado, you know, that was sort of the bio, I guess, on your NIH bio sketch. But we want to hear from you. So, you know, you're the Director of Internal Operations at the Project ALS Therapeutics Core at Columbia. What led you to that position? How did you become the overseer of all of the science at the Core?
Well, yeah, just to be clear, there are co-scientific directors who are my bosses, and they, you know, have some say in what we do. But, yeah, I guess, you know, a lot of it started with work in the Jennifer Estes lab at Columbia and Project ALS, and you know, the work there. The initial project that I started there wasn't super related to drug discovery, but that project actually didn't pan out, and I got lucky and joined a project that was on drug discovery that somebody was leaving behind. So once I took that over, it was just a really good fit for, I guess, the way that I like to work and the way that I like to think. And so yeah, so that, like you mentioned, the work on prosetin has taken a few years, but that's because there were a lot of hurdles to overcome in developing that screening platform. And yeah, so I guess, you know, as Project ALS was looking to sort of pivot towards the funding, not just of basic science, but also of drug discovery, the work that we were already doing together sort of became a natural fit for the work in the Core. Yeah, I guess on top of that, you know, I had a relationship already, obviously, with the scientific co-directors. So Henrik was already my boss, and I had a good relationship with you guys. And I knew already some of the major, the families that were helping to donate a lot of money to get the Core up and running through the drug discovery work that we were already doing. And yeah, I guess it just seemed like a natural fit, hopefully.
And obviously today, Emily is not broadcasting from the lab, that's a scientific lab, because academic research at Columbia outside of a few specific things has been paused during the COVID-19 pandemic. But, you know, on a day where you would be in the lab, what does sort of the day-to-day role and responsibilities of running the scientific operations of the Core look like? Yeah,
so it varies day to day, but usually I'd say a lot of my time is devoted to working with the technicians who really get the, you know, do the day-to-day science in the Core. So they handle the mice and they feed the cells and, you know, just make sure that everything is is going smoothly. And, you know, there's also a lot of technical work that we're taking on that requires a lot of training. Yeah, we spend a lot of time together just learning new techniques and making sure that everything is working smoothly. And, you know, setting short-term and long-term goals with them to make sure that everything stays on track. Yeah, I also spend a lot of time fighting with my bosses about new experiments and things like that we want to do. And, you know, everybody has a different perspective on what's important in ALS and drug discovery. And it's not really fighting, it's, you know, educated discussions and that are very polite, but sometimes, it's a really good way when people disagree about, you know, what's important in ALS, then you sort of, it's a good way to eventually hone in on what is important if you can convince everybody else in the room who's skeptical. So that's another big part of my day. A lot of meetings, a lot of emails, a lot of scheduling. Erin, I know that's a big part of your job too. We try to together sort of, well, Erin initiates it and then I try to follow up, like chase down other scientific collaborations outside of Columbia. Again, because, you know, there's a lot of really smart people at Columbia, but there are other people in other institutions who have really good ideas or really good drugs, but they don't have, you know, the platforms that we do to be able to test them. So we try to set those up, and that takes a lot of emails.
So just for those who may not know, and Erin, maybe you could also provide a thumbnail sketch of the core. The core was put together in order to test other promising drugs, not only drugs that come from Columbia scientists, but drugs that come from drug companies and biotechs and other academic labs. So our goal was to set up a system, kind of an obstacle course of testing at the core so that we could run virtually any therapy through that. But what really strikes me about prosetin is that Emily and Erin, when it popped for you guys, we were able to partner up with other companies that have just become part of the team. So it's suddenly become a bigger, more dynamic team.
Mm-hmm. Yeah, no, and you know, I think sort of to that point. So as Valerie said, you know, the idea behind the core was that there were promising, you know, therapeutic targets and even potential therapies that were emerging from Columbia-based research. And then we at Project ALS also saw from other academic groups that we fund and industry groups that were now interested in ALS, all of the sort of promising genetics and biology that could lead to meaningful treatments for ALS. But there was this gap between promising new science and then what was actually available in the clinic for people with ALS. And as Valerie said, a big part of the core was to put this sort of obstacle course basically in place by which Columbia-based researchers or anyone else could evaluate, you know, a potential drug in a really thorough, efficient way, and have a really strong scientific rationale when you then move to the clinic and to people with ALS that this drug had a good chance of working. I'm kind of wondering from your perspective, Emily, because this is sort of a new initiative. It's not something that Columbia has really done before, although there was a strong expertise in basic ALS research and then in ALS clinical trials, but this sort of brought everyone in ALS research at Columbia together to work in a more streamlined way, maybe in a more drug development oriented way. So are there sort of opportunities or changes that you've seen the core bring, you know, to you and to the general group or to Columbia ALS research in general?
I think, well, yeah, so from a scientific perspective, I get to work a lot more closely with, even though we're all at Columbia, we don't necessarily all talk all the time. So it's an opportunity to regularly get people in the room who think about different sort of aspects of ALS and can all inform, based on all of our experiences collectively, sort of inform new experiments. You know, what if somebody's already tried something and it didn't work, you know, we can think about why, and or if somebody has a really great idea that others of us might not have had because of our different backgrounds, that's also super helpful. But I think, you know, another really, not to keep talking about money, but I think like a really big factor in the core, I think, being successful is the fact that we don't thanks to the generosity of Project ALS, we don't have to sit in our labs all day writing grants. And that is, as anybody in the field can tell you, like just a huge time suck. Like it takes months. You're proposing, like there's a lot of restrictions on what actually will get funded through things like the NIH. And even though those are really, really important funding sources for science, and there's still a lot of things that they won't fund. So they won't fund really a lot of drug discovery because you can't guarantee that you're going to get a good result. So those kinds of things that we do are really difficult to get traditional funding for, even though there's obviously a huge need for ALS drugs. So the fact that Project ALS sort of took that huge barrier off the table has allowed things to move really quickly, and to move in a way that where we can be a little more open-minded about the kinds of things that we test.
Yeah. So now, I mean, I think that was sort of the idea behind the core. Now, prosetin is the first in-house drug candidate from the core that's actually, you know, emerged as a top performer that we're trying to take to trial. And as Valerie said, that was a whole other shift where we had to bring together this new team of external partners, people that academics sort of never work with, manufacturers like Patheon and contract research organizations like Charles River, people, toxicologists and regulators and CMC experts, you know, oh my.
Yeah.
So, you know, what makes prosetin development different from what your prior day-to-day was? Is there another shift sort of in how you think about, you know, what science looks like in the context of drug development versus just screening drugs in a lab?
Yeah, it's really different. And I feel like it's gonna make us sound bad, but you know, so we laid the groundwork for prosetin, you know, by like trying a lot of different versions of it before we came up on the right one, and we developed the right tests to make sure that it was working or not, because that's, you know, how would you know if it worked if you didn't have the right metric? So like all of that stuff was stuff that we did in-house, and I think that that's really best done in an academic environment where people are thinking really critically about outcomes and biological pathways and things like that. But in terms of assessing, you know, the second step of getting something to a patient is making sure that it's safe, and that's something that I'm realizing thanks to our interactions with everybody, the huge team that's been assembled by Project ALS. We don't really have the capacity to do that kind of stuff in-house because it requires, you know, a level of oversight and care that we're just, you know, this is something I didn't even know about at all. So like, you have to record the serial number of the scale that you're using every day to weigh each mouse and each drug, and, you know, it's stuff like that that's like, not to suggest that we normally take shortcuts, but
no, but I think, Emily, you raise a really key point, and this is what separates Prosetin 2020 from other efforts that I've come to know over the years. You and your academic partners bring ALS expertise, disease, you know, knowledge about disease pathways, as Erin said, new genes that have been identified as role players over the years. And you kind of do the heavy disease lifting and create the platforms for discovery. But when it comes to going online with a drug, we need other partners, people with expertise in other areas. So how do we take this sort of great looking academic product online for people with ALS? And that's why, you know, when I first introduced Erin to introduce you, you were really there at the beginning. You're the scientist. You bore this fruit. That sounds weird, but but now the Prosetin 2020 team is all working together to help figure out how can we get this jewel to more people. And that's where you mentioned Charles River comes into it, and the many consultants who comprise Prosetin 2020. And of course, Patheon, which is a company, Erin, you probably put it better than I, but Patheon is a company and our partner in getting Prosetin to the people who actually manufacture drug. So I mean, Emily's in her home right now. You don't want her making the prosetin that, you know, eventually will go to human trial. It's a bad joke, but so prosetin is being manufactured in proper ways, responsible ways.
So, so
can I just add, sorry, I just wanted to add that, Valerie, the number that you quoted at the beginning to get prosetin to human trials, like $1.6 million, seems maybe large for a lot of people, but you know, it's something that is untenable for an academic lab. You know, if we came upon prosetin by ourselves and we didn't have access to that kind of funding, it would be dead in the water, basically. There's no way that we could have raised that money, and there's not really a lot of outlets, again, from like traditional funding routes for academic labs to move that forward. We are pursuing some of them just to make sure that we really get as much funding as we can for this to move it forward into patients. But yeah, so normally, you know, that's why a lot of drug discovery is really restricted to big pharma. And Erin, as I think you've definitely said on other webinars, like the drugs that they choose to pursue are ones that have been deeply de-risked. So they're looking for things that are going to give them a big return on their investment, and you know, all that stuff makes sense from a business perspective, but they're not necessarily prioritizing things that have a very strong scientific rationale and the opportunity to help a lot of patients. And we can talk about what that means later maybe, but yeah, I just wanted to drive home that this is a really rare situation in drug discovery where something is coming out of an academic lab, doesn't have to go through big pharma in order to get into patients.
But don't you guys think that if successful, and we plan to be very successful, that this could provide a template going forward for disease experts to do discovery and then pass the baton to commercial partners?
Yeah,
that's I guess reason number 4 why I'm so excited about prosetin, because it's a marriage between academic efforts, nonprofits, patient advocacy, and commercial efforts and expertise.
Yeah, I think my sense from talking to people who are in drug development in the pharmaceutical industry, they kind of initially didn't take our efforts very seriously because they're like, oh, like you guys are so little. Like, what if you find something? What are you going to even be able to do with it? But I think you know, there's sort of, especially after, you know, we were able to publish our work and things like that. I think people are taking it a little more seriously and understanding that the platforms that we're using for drug discovery are things because they were developed by, you know, the experts in the field, like, you know, my boss who discovered how to make motor neurons in the first place. You know, we're doing a kind of drug discovery that isn't possible in pharmaceutical settings, in my, you know, in my view. But yeah.
So let's talk about these platforms a little bit. And, you know, we don't need to get deep into the weeds of scientific discovery, but I think this is a really basic question in ALS drug discovery. So, you know, Project ALS and Columbia and other collaborators, most significantly Harvard, invested really early in the idea that stem cells could be a really powerful tool for ALS drug discovery. And back when we were involved with, you know, when we funded the Project ALS Laboratory for Stem Cell Research in partnership with Columbia, a team based at Columbia with your boss, Harvard, Kevin Eggen, and the Project ALS Lab, developed the first patient-derived stem cell models of ALS. And the whole idea was that you could make these motor neurons from people with ALS that had their disease. You could put them in a dish, you could screen thousands of drugs, and you would just know for any individual patient what drugs could help rescue their motor neurons with ALS. This was Time Magazine's Scientific Breakthrough of the Year in 2008, and it's very impressive, but it's also sort of disturbing that in 2008, that was the Scientific Breakthrough of the Year, and we still don't have, you know, really effective drugs approved for ALS. So maybe you could talk just a little bit about sort of what the challenges were with that platform, and then how you and Dr. Victorly and other collaborators at Columbia worked to improve that platform so you could actually use these patient-derived motor neurons, this ALS in a dish, to screen for drugs.
Yeah, yeah, and I just want to say that it's like a continuing process where I don't think we've fully solved any of these problems, but So the process that Erin is talking about to get patient cells and develop them into motor neurons. So first you can take a little bit of a blood sample, or you can take a skin sample. So, in the instance of like skin, you would take just a little patch out of your skin. And the cells in your skin are called fibroblasts. And then you can take these 4 reprogramming factors, and that won the Nobel Prize in, I don't know, I think 2013. That's probably wrong. I think it was 2011. They're called Yamanaka factors, and they basically you only need to turn on these 4 genes in order to make a skin cell or other types of cell turn back the clock and become a stem cell. So once you've gone from skin cell to stem cell, the stem cell is something that I'm sure everybody has heard about, but it's especially promising because it can become any type of cell as long as you give it the right instructions. So my boss and other people originally just figured out what those instructions were to make a stem cell become a motor neuron. So once you sort of have figured that out, it's actually really difficult. It was really easy to do for in the early versions where all this was being done with mouse stem cells. Just making a mouse stem cell into a motor neuron took like 2 steps and took a week. But somehow the discovery that Erin is referring to was the process of being able to take a human stem cell and make that into a human motor neuron. And that was actually much more complicated and time consuming. And that took years to parse and figure out. So once you're able to plate stem cells from a human patient and make those into motor neurons, the expectation was like, okay, great, we have, motor neurons are the cells that when people have ALS, they get very, very sick. Those motor neurons are especially vulnerable. They're the ones in your spinal cord that tell your muscles what to do. So if they die off, your muscles don't really have instructions anymore. By the time that patients are symptomatic, they've already lost, 50 to 60% of their motor neurons in their spinal cord. So we just everybody assumed that if you were able to make these motor neurons of very sick people, that the motor neurons in the dish that you're looking at would also be incredibly sick, because we know this person had ALS and they're very, very sick. But the sort of open secret in the field after that 2008 paper came out is that the motor neurons that you make out of iPS cells, stem cells from patients, not that sick. Maybe if you look hard, you can see like a 5 or 10% decrease in survival compared to motor neurons that were made from healthy patients. But that's not enough to see the effects of a drug. When we put together drug screens, we like to see 50% decreases in survival. So then you have a lot of room to find drugs that do a little bit of good, a lot of good, somewhere in between. And so that's that's the whole range of detection that you want. You want that to be big. So I think everybody was disappointed and surprised when that discovery was made, that they're not really that sick. And there are a lot of theories as to why that happens. One theory that I think is maybe at least partially correct is that when you take something like a skin cell from a patient and you make it into a stem cell, somewhere during that process, you strip off all of the markers that tell a cell how old it is. So you basically take a cell from, people have done this, you can take a cell from somebody who is 100 years old, take a skin cell, transform that using those Yamanaka factors into a stem cell. And then the stem cell, from a biological perspective, looks like it's the stem cell of a baby. So you took 100 years and you erased 100 years off of the clock. And why that's important for something like ALS is that most people don't develop ALS until they're, in their 50s or their 60s. So if you're looking at the stem cell derived motor neuron of a patient who didn't get sick until later in their life, you're looking at a stem cell, a snapshot of a motor neuron from a period in their life when they were healthy. So that you're not really recapitulating the effects of the disease, and one of them being aging, which is a really important factor here. I think some of the other problems are that motor neurons, when you look at them alone in a dish, in your spinal cord, they actually live with, they have a lot of neighbors, and many of them are not motor neurons. They have a lot of support cells, they have a lot of immune cells, they have a lot of sort of other intermediate cells that, that interact with motor neurons every day, and you don't really recapitulate those interactions in a dish. So I guess those are sort of the major hurdles that are still being overcome. There are a lot of different theories as to how you can do that. There's some people who suggest that you can go straight from a skin cell into a motor neuron without having to go through that like age erasing process in the middle. So that's something that we're thinking about. But I think really the practical solution that my boss came up with another postdoc in the lab who has since left, but he's great, Sebastian Tams. So they figured out like, okay, there's probably some, we know eventually that these patient stem cell derived motor neurons would eventually get sick if we were able to grow these cells in a dish for 60 years, for instance. Eventually we'd be dead, but eventually the motor neurons, the theory is, would develop a pathology. But the assumption there is that there's maybe something going a little wrong early in, even when you're a baby. Like maybe it takes a long time for the effects of what's going wrong to accumulate until the point when you finally get sick. But the idea was basically to try to find a way to exacerbate anything that was like a little bit wrong. So they developed, the initial screens that they were actually doing were not for protective drugs. They were for anything that could like exacerbate that small difference between healthy patient and ALS patient stem cell derived motor neurons, and try to really shift that difference in survival between them. And so they came up with a few drugs actually that were, so these are stress drugs that were able to, they killed ALS motor neurons more readily than healthy motor neurons. So they were exacerbating something that probably was already wrong in the ALS motor neurons, but that we couldn't see yet. The thing that they were exacerbating that Valerie mentioned early on, and which is what we're trying to target with prosetin, is a process called ER stress. And sorry if I'm just going on and on, please stop me if it's too much. But so ER stress is something that happens when, and motor neurons are very vulnerable to it. So it's when they see misfolded protein, like if a motor neuron sees misfolded protein, or if it sees like an accumulation of proteins that it wasn't expecting, they really freak out. They're like, no, I told you I did not want any more misfolded protein. I can't handle it. I don't want to tell you again. And then, so finally the motor neuron gets so overwhelmed and upset that it kills itself. So which is it's actually a protective mechanism, because the motor neuron doesn't know what it is. So in order to prevent whatever is going on in the cell from spreading, it tries to shut itself down so that it doesn't get worse in the rest of the spinal cord.
Just to summarize, what I hear you saying is that just putting stem cell derived motor neurons, even if they come from people with ALS, their skin or blood, do not really show the disease state in the dish. But what you have done with your colleagues at the Core is created a platform in which you subject those motor neurons taken from patients and stem cells to stressors. And so you sort of approximate the types of cellular stress undergone in ALS. So you put them in that environment, and then the motor neurons in the dish sort of become ALS in a dish.
Exactly. Yes.
Is that, am I following you?
Yes, thank you for putting that more succinctly.
No, I mean, so that's my job. We want everyone to understand this process, and what you've done is so exquisite. I mean, you've pretty much approximated ALS in a dish. And so what we do at the Core now is that platform is used to screen for drugs. So if you can approximate ALS in a dish,
Yeah.
who's to say, you can't throw drugs into that dish that will rescue motor neurons. And I believe that's where prosetin comes into the, makes its way into the headlines, because, tell us about that, Erin and Emily. Prosetin, more than other drugs introduced to that dish of sick motor neurons, protected them and even rescued some motor neurons that were not doing well.
Yeah, I think I would just say from, my lay perspective that, first of all, it's kind of amazing. Well, first of all, I think, Emily's explanation of these motor neuron models is really important, because we have this idea of ALS in addition being able to readily screen drugs. But it was very difficult, I think, for this team of experts at Columbia to get to that point. I think what's really amazing now is at the core, because of all of this work that Emily has described, this is like a bread and butter thing that we can do now. So it's very easy. It takes a couple of weeks to make these stressed out ALS motor neurons and to screen thousands and thousands of potentially therapeutic drugs on them. And we can see which drugs actually rescue ALS motor neurons. And again, because of all the work that went into developing this cell screening assay, this way to screen drugs in ALS, we can say really confidently that prosetin is, the top performer out of all of these years of work. Have brought us to prosetin as a drug that rescues these stressed out ALS motor neurons, calms them down, makes them healthy and happy and able to function in this stress assay.
Yeah, and I'll maybe also just add that actually when we first started doing this kind of screening, like where Valerie said, we're sort of recapitulating a stress that already exists in ALS patients. We're just sort of amping that up in the dish. It's actually not that rare to find a drug in the dish that works. And if anything, we're finding more drugs than we knew what to do with. The problem was that none of those drugs were really ready to put straight into patients. And that's really why something I didn't understand when, we first started out. I thought this would be, like a one-step process. You just find something that you could just order and then put into patients. But yeah, so something that we spent most of our time doing actually was taking the parent version of prosetin, the original version, that worked really well. Like Erin mentioned it. It basically didn't allow any of the motor neurons. We were trying to kill half of the motor neurons in the dish, and it didn't let any of them die, which is a huge result. But it just wasn't in any shape to put into patients, and it didn't really get into the brain very well, which is where you need to go when you have a brain disorder like and a spinal cord disease like ALS. And we also found that the drug was broken down really quickly by the liver. So even if we were able to get it into into the bloodstream and into the body, as soon as it hit the liver, it would be gone. So there are a lot of reasons that, again, this is something that I didn't even really understand was a part of drug discovery, that took way longer than finding the parent version of prosetin itself, was to make it the best version that it could be to get into a patient with ALS. So that was another thing that we really spent a lot of time thinking about and doing. And now, because we had to go through those hurdles once, we are able to move a lot more quickly through that process for subsequent drugs. So we're thinking about other compounds now, not just prosetin, and how to streamline that process. So we're not just, spending like 4 years basically making a stable version of a drug. Now we know where to look, basically.
Yeah. So we have a couple of questions, and if anyone else has questions, type them in, but maybe we'll just answer 2 or 3 now before we send Emily off to a scientific meeting that I know she has. So here's a good one. Is there any possibility that prosetin could have applicability to other neurological diseases besides ALS?
That's a great question. And I think that yes. We've actually done a very some preliminary tests in, we've given prosetin to our colleagues who do other types of neurodegeneration research. So like one example is somebody who works with Alzheimer's disease, and we gave prosetin to them to see if it worked there. And in their model of Alzheimer's in a dish, it looked really good. And they actually said that it looked better than a lot of the drugs that they use as a positive control. We also gave it to some people who are looking at it for chemotherapy-induced peripheral neuropathy. So when people take chemotherapy, sometimes the nerves in their their digits, their hands, and their feet start to die off, and it causes a lot of pain. So there's a model of that in addition, and prosetin looked good there too. So I think as we discover more about how prosetin works, we're discovering that it targets a really a mechanism by which a lot of types of neurons die, basically. So, it's important for motor neurons, but we're finding that it's also important for the kinds of neurons that are vulnerable to Alzheimer's disease. ER stress is also just a really common thing that happens to a lot of cell types, not just neurons. So we think we've hit upon a pretty common stress pathway that, if we can block that, could be helpful for a lot of different cell types and a lot of different disease contexts.
And then another question, which actually kind of looks forward to the next part of the series: what is the projected time from trial to market? So I'm actually going to put a pin in that question because I think in our next webinar, we're going to be hosting the clinical trialist and ALS clinician, Dr. Jinsey Andrews of Columbia. And, we want the experts to answer that question, not me. So maybe, Valerie, do you want to look forward to what's happening in the next?
Yeah, let's all look forward together. I really want to encourage you all to participate in these live streams. You ask great questions, and we want to get to know you. So one of the ways to do that is to continue our live streams on a weekly basis. So next week, let's see, I believe it's April 28th, according to my calculations, we will meet Dr. Jinsey Andrews, who is the ALS neurologist who will oversee the first trial of prosetin in ALS. We're very excited about that. And then the following week on May 5th, we will host Dr. Lauren Black of Charles River Laboratories. She is a distinguished scientist with Charles River. And again, Charles River is part of the prosetin 2020 team. They're helping us to cross all of the T's when it comes to getting prosetin safely to patients. You don't want to do further harm. And Charles River is leading us toward safety with prosetin. And then I would also mention, this is kind of a sidebar, but back to the ways that we all can help each other, help prosetin 2020, is on a date to be announced. It'll be soon, probably toward end April, beginning of May. Project ALS is hosting online Women and the Brain, which is a really fun night. That stars comedian and Emmy Award winning writer Judy Gold. It's an evening of education, fun. I would go so far as to say debauch. Okay? But it's fun. It's a good way to kick back for all of us who have felt additional stress these days. But P.S., it's also a very effective fundraising tool for prosetin 2020. So we'll see you at Women in the Brain. We'll be providing all the details so that you can participate at the push of a button, a click of a switch, whatevs. But in the meantime, guys, we really need you. We need to keep Emily on track. We need to keep Charles River and Patheon and all of our brilliant consultants and Erin herself, for heaven's sake, on track to get prosetin to its first patient trial in 2020. We're still gunning for it. Please go to prosetin2020.org. That's prosetin, Prosetin, 2020.org. $5, $20, $100 for you fat cats. It's been an honor, Emily, hosting you today.
Thank you.
Thanks for all you've done to bring prosetin to reality. Erin, as always, great to share a computer screen with you. And we're signing off for now. Please stay in touch with Project ALS, stay safe, and let's go prosetin 2020.
Bye, everyone.
Bye.
Bye-bye.
Thank you. It's been an honor.